the different nanoparticles that can or should improve the wider use of mRNA to treat and prevent various forms of cancers. What are they? Can you talk us through that?
Yeah, so I mean, so there are sort of broad categories for it, right? So there's lipid nanoparticles, which are again quite common, and they have a cationic lipid in them, and but a lot of work has actually gone into figuring out the structure of lipid nanoparticles that make them, one, deliver, but two, are also safe. The early generations of lipid nanoparticles for delivery of messenger RNA were quite inflammatory. They had, safety concerns. So that a lot of that's been dialed out through a lot of work by a lot of different companies and academics and so on. That's sort of your fundamental foundation for the delivery. The polymers, more work has gone into using polymers, but again, sort of the same thing where you are forming a particle where the polymer is a component of that particular particle. The issue with messenger RNA, even above and beyond other oligonucleotides, is that messenger RNA is very long. An siRNA is 20-ish nucleotides long, and antisense oligo about the same. Messenger RNA is obviously kilobases in length, and that limits what you can do, the changes you can make to the messenger RNA itself. In the case of siRNA or antisense oligo, you can essentially modify every single nucleotide present in those duplexes and provide it with drug-like properties. There is a much more limited way that you can do that to messenger RNA, and one, still have it be functional, and two, even physically do that, because you're not chemically synthesizing the messenger RNA. You have to you're essentially in vitro transcribing it, which limits what you can do to it. So there are innovations that have been used in the short oligonucleotide space where you can directly conjugate ligands onto the siRNA, or conjugate it to an antibody, that sort of a thing. It's much more limited in your ability to do that to a messenger RNA. You're essentially you need to make something that can encapsulate it, a nanoparticle of some sort, and then essentially provide targeting ligands onto that nanoparticle to sort of adjust where it can go from that point. But there are a relatively, at this point, limited number of things that have worked to actually deliver in vivo. Lots of things work in vitro, and that's what was being discussed earlier. There are many ways to get RNA for ex vivo applications. In vivo is much more difficult, more limited, because you're trying to solve multiple problems at the same time.
Okay. I think a question that was submitted by an audience member is probably applicable now. And this person is asking, what are the key reasons for less than 4% LNP mRNA effective inside of the cells?
That's because it's death by 1,000 cuts, essentially, is what it is when you take the lipid nanoparticle and you encapsulate the messenger RNA. That's great. You've got a particle but it's essentially then needs to overcome all of those barriers that I was mentioning before. So some of it's going to get destroyed as soon as you put it into serum. The particle itself will be destroyed. Even if you can get it to, and I'll use the hepatocyte because that's obviously where lipid nanoparticles commonly traffic to. When the LNP fuses with the cell membrane and the cargo is released into the endosome, it's then trafficked through that process as well. So some of it is just barely able to escape from the endosome during that maturation process, for example. Some of it can be actively transported. A lot of it is destroyed as the endosome essentially matures into the lysosome, and what's inside of it, your messenger RNA, is being destroyed as well. So you're basically, you're in all of this delivery, you're essentially trying to overcome those systems that are trying to destroy it. So the fact that you're even able to get 4% is terrific, because so many things are working against what it is that you're trying to deliver.
Okay, thanks. So we've talked so far, we've referred to cancer oncology indications. What I want to talk a little bit more about, Emirat, I'm going to direct this question to you because Cartesian has therapies outside of cancer. And I was hoping you could talk us through what the RNA therapeutics are for your respiratory and autoimmune therapies that you have going through the clinic now, where we are with that, and from a delivery perspective, can you bring us up to speed on what's going on?
Yeah. So, yeah, and I want to phrase my comments in a way that I think would maybe borrow from how Walter sort of teed it up, and from really a sort of a broader perspective, because I think that there's a lot here, and so again, I just want to emphasize that when we talk about RNA therapeutics, that the conventional way to think about it has been to date delivering RNA either sort of by itself or in the context of some carrier, inert carrier molecule, a lipid nanoparticle, for example. But RNA therapy is much broader than that, right? And there's different ways to solve the delivery challenge. And the way that we've gone about it is to use the cell as both the factory for production of, as well as the vehicle for delivery of one or more different RNA therapeutics. So let me just sort of even take a step back and just discuss why this problem was interesting to us in the first place. So we started off back in 2016 in an attempt to deliver potent yet safer cell therapies. And we took a program, a CAR T program that was traditionally being developed to treat cancer. And the conventional way to engineer cells is, of course, with DNA, right? You're making irreversible permanent changes at the level of the cell. And we thought, well, can we make a CAR T, but make it much safer so you don't have to worry about all the toxicity associated with it, cytokine release syndrome, neurotoxicity, risk of infection, risk of transformation, et cetera. If you're not asking your nucleotide to get into the nucleus, but rather stay in the cytoplasm, if you're substituting the DNA with RNA, you have that ability to predict the length and the level of expression of that RNA. And so as a result, you gain a lot more control inside the body in terms of what your RNA therapeutic is capable of doing. And so we went about that mission, and the first product that we developed ended up being an R-CAR T, if you will, an RNA CAR T that was targeting now a very popular and common target, a BCMA, which is ubiquitously expressed in plasma cells, as a way of targeting at first multiple myeloma, but also long-lived plasma cells that are now a hot area of exploration in autoimmune disease and neurology. So we generated an RNA CAR T that targeted this antigen, and began this journey of moving that program into clinical studies. Now, fast forward several years, with that lead program, we've not only defined the safety profile of this RNA cell therapy as being incredibly safe, no evidence of cytokine release syndrome, no evidence of neurotoxicity in dozens of patients that we've treated, but we also have now leveraged that safety in order to take it beyond oncology into a classic autoimmune disease and neurological disease called generalized myasthenia gravis. That's a disease that affects the junction between the nerve and the muscle, and as a result, patients become incredibly weak over time. Despite many years of of attempts to create better and better, more targeted therapies, the mainstay of therapy for myasthenia remains broad immunosuppressive and highly toxic therapies. So there's a real need for more selective therapies. And so we've been at this and are currently in a phase 1/2 study, and just recently presented some interim data from the phase 2 portion of the study. And there are remarkable responses to the point where we are seeing deep and looking like durable remissions in these patients, to the point where there's really no evidence of disease that's detectable, and very exciting, very early and yet very exciting data, combined with an incredibly promising safety profile that is just not typical of cell therapy, the conventional cell therapy, right? So that's one example of how the cell can be used as a factory for producing as well as a vehicle for delivering one or more different RNA therapeutics, is this sort of program. And we use that we used an example of a T cell where we isolate from the patient, engineer the T cell and give it back to the patient. In the process, we can overcome the delivery issues, the transfection issues, et cetera, because we are by definition getting it into the right cell before even infusing it back into the patient, right? So that's one example. Now I'll just quickly give a second example of a different type of cell we work with, a stem cell, a mesenchymal stem cell, an MSC, where you have the opportunity to use these types of cells as an off-the-shelf or allogeneic therapy. So in our MSC programs, we take umbilical cord-derived mesenchymal stem cells, take them out of obviously a healthy individual, and then engineer them outside the body with 2 or 3 or more different combinations of RNAs, and then administer them back to a patient. And we have programs not just in oncology, but also in respiratory disease, ARDS, and severe pneumonia, and hopefully soon a local indication in diabetic foot ulcers. So the cell is very special in its ability to avoid immunogenicity, right? In its ability to be engineered to carry that combination directly to the site of disease. So one of our programs, we're actually introducing a homing signal onto the cell membrane in order to deliver its cargo right to the site of disease, as well as its capacity to keep producing the protein at substantial levels when the RNA goes into it. And of course, its ability to carry multiple RNAs is, I think, a key differentiator relative to the cargo limits of what you can expect to get into a nanoparticle. So we think that there's a lot of advantages above and beyond solving for the delivery issue. And we're excited at the prospect of pioneering this new class of therapies as a way to try to solve some of these issues that we've been talking about.
Yeah, for sure. And it sounds like absolutely there's great data to be celebrated there. So please keep us posted on how that continues to go. Absolutely. One of the things I want to talk about, and Walter, I think this probably would be a good question for you. One of the concerns, and we've talked about it a little bit, but I want to get into more detail. One of the concerns for mRNA therapies is that they can be difficult to obviously deliver and dose effectively. Talk to us about the advancements that have been made with delivery stability. That's a pretty broad topic, but
yeah, no, that's great, and it's so when we've been talking about messenger RNA here, obviously we've been focused on sort of what I'll refer to as the conventional messenger RNA, which is an RNA that looks very similar to what it is that's naturally occurring when our cells make messenger RNA. Obviously, there have been significant advances around making circular RNA, and that offers significant advantages depending on what it is that you want to do. So a messenger RNA that's linear, first of all, it's exposed at both ends, so it's going to be more susceptible to what I was talking about before, the nucleases that are targeted to destroy it, and to mechanisms that are designed to detect conventional conventionally formed messenger RNA. The circular RNAs, and there are two companies that are predominantly working in this space, Laurent and Orna, and some may even be on this call right here, but those offer certain advantages in that they are not as susceptible. And the other part of it is that you can make more protein off of those messenger RNAs because they exist, because they stay around for longer in the cells. So if you want to do something that requires a more sustained expression of a particular protein, modifications like making these RNAs circular are a significant advantage. So if you go back to the vaccines, for example, you could argue that you actually want transient expression of the spike protein of COVID in the making of a vaccine. You don't want it to go on for too long. Though what the optimal length of time is, I don't think we really know, and circular RNAs may be beneficial in that case as well. In the case of, more broadly speaking, what I'll call sort of refer to as enzyme replacement, where you want to make a particular protein for an extended period of time. Again, circular RNAs may offer advantages in that space as well. If you are talking about linear messenger RNAs, then obviously there are modifications to those nucleotides that can be incorporated during the in vitro transcription process, but not to the degree where you're talking about a short RNA where you can chemically synthesize it. But again, those modifications can help adjust. They can allow you essentially to tune up or down the level of expression that you're getting, the length of time that the RNA exists, which can be optimized for the specific use case that one has.
Okay. Let's talk about protecting the mRNA cargo against degradation. How does a well-chosen delivery vehicle, right, protect the mRNA cargo against degradation or immune recognition without the need for this extensive chemical modification?
So sure. So again, it sort of goes back to the very beginning, that you're starting, from the outside and you're working on your way in. The delivery vehicle has to essentially prevent the RNA from being seen by the nucleases from the system that exists within the blood. Once the messenger RNA is actually delivered within the cell, essentially, and then the lipid nanoparticle is not relevant anymore. Essentially, the cargo has been released and it's exposed to an intracellular environment. You do need some degree of modification to the messenger RNA in order to have it survive for a long enough period of time. So the first step is you need to be able to encapsulate that RNA to protect it until it can at least get to the cell that you want to get it to.
Gotcha. Okay. What are your thoughts on the conjugation of other RNAs to lipids and polymers?
So the issue there is that you can do it. You can conjugate lipids and cholesterol and whatever else, and GalNAc and so on, targeting ligands to a messenger RNA. The issue is that as soon as you put that, if you inject it into circulation, the RNA is going to be destroyed by nucleases. Then that's what they exist to do. The way that you're able to overcome that with the short RNAs, again, is because you can introduce such extensive modification, you can essentially make them very resistant to those nucleases. That's not really something that you have the capability to do simply by conjugating, a few lipid groups or whatever onto something that's kilobases in length.
Okay. Leading up to our event, like I said, we had several questions and topics that wanted to be covered. And one of them was, what are the unexpected uses for mRNA delivery? What are your thoughts on that?
I'm not sure what unexpected means. And since it was discovered that mRNA could be delivered, like you could make mRNA and deliver it into cells. People have had lots of ideas about what it is that you can do. The vaccines, everyone is familiar with that. Some of the work that's been done with gene editing perhaps might be one of those unexpected uses.
Right.
Essentially, there you've got Cas9, and that is, for example, if the work that's being predominantly done at Intellia is to make the Cas9 as a messenger RNA and deliver it by lipid nanoparticle, not because you necessarily, the Cas9 itself is not therapeutic, but it allows you to edit the DNA and have a therapeutic impact by doing that. Again, it's done transiently. It expresses Cas9. There's a guide there as well. It does the editing event, and then that editing event is, so far it appears to be, permanent and heritable by the cells that you've done the changes in. So that is perhaps slightly different than what was originally thought of, where you take an mRNA from a virus for vaccines, or where someone is deficient in a particular protein and you provide it in the messenger RNA form. So perhaps the gene editing piece of that would be an unexpected way of leveraging the messenger RNA technology.
I mean, I will say that in general, there has been a lot of promise in the field to treat specific diseases with nanoparticle delivered RNA that showed interesting, almost exciting data preclinically, that when it moved into the clinical setting, it just didn't pan out. And I think a lot of the reasons why the RNA vaccines have been so effective is the reason why a lot of the RNA therapeutics have been not so effective to date, right? And it comes down to immunogenicity and targeting, right? It's as Walter said, there are systems in the body that are designed to recognize these treatments as foreign. They mount an immune response towards them. They try to clear them out. In the process, if you can get some degree of expression in the context of an immune response, then that's like the perfect combination for an RNA vaccine, right? But the very nature of that potent combination for RNA vaccine is the exact reason why you don't end up getting a lot of clinical efficacy, especially after repeat treatments with the conventional nanoparticle-based RNA therapeutics. So, trials from, I think that treating liver diseases might be somewhat of an exception here. But when you're targeting diseases that, are systemic, or need local delivery following systemic infusion, such as treating respiratory diseases, for example, it's very tricky. It's very difficult to A, target these cells to the right place, and B, do it in a way that doesn't wake up the immune system and get it to clear.
Yeah, I agree with everything you said, Murat, there. That's exactly right. One, the vaccines are the best possible use case for messenger RNAs. And yeah, then getting it into the relevant cell it's not an insurmountable issue, but it is a very, very difficult problem. To get messenger RNA into something that's not, dendritic cells or hepatocytes it's a very difficult thing to do.
Do you see the potential of mRNA-based therapeutics in autoimmune diseases? And what would you say would be the right delivery platform for autoimmune diseases?
Let me give I'll give a broad answer to that, and then Murat maybe can drill into a little bit more detail. So I would say it probably does have relevance for autoimmune disease, but it's going to depend on precisely what autoimmune disease you're talking about and what it is that you need, what therapeutic intervention you actually need to engage in. So each of these diseases is going to be different, and the way that it manifests is different, and the target cell arguably is going to be different as well. So it's not something where you can say it will definitely have relevance for all of these. Again, it will be use case specific. It'll be therapeutic intervention specific.
So I would say, Walter, I would say yes and no to that. So I think yes, insofar as your target is specific to an indication that differentiates it at a very core level from another indication within the class of autoimmune diseases. But no, insofar as your targets a node in the mechanism that is common to many autoimmune diseases. So, our experience is that the long-lived plasma cell that is actually generating the autoantibody is a ripe target. So these cells are called long-lived because they're around for decades. And it's difficult to target them effectively. And so what we try to do in our autoimmune programs is to do just that, to go and target and find and eliminate these pathogenic autoantibody producing long-lived plasma cells. And we believe that if we can do that with our first indication, myasthenia, which are generating these autoantibodies to acetylcholine receptor, to the MuSK receptor, and other sort of aspects of the neuromuscular junction. If we can eliminate a pathogenic autoantibody producing cell in that indication, we certainly can do so for a whole array of other autoimmune diseases that at a very fundamental level have the long-lived plasma cell as its node. So, immediately the options are going after lupus or neuromyelitis optica or, a whole array of diseases, pemphigus, start to open up where the LLPC is the likely, at least in a key, if not a central node in the mechanism of action.